Hi, my name is Deirdre McLaughlin, and I am presenting our poster on behalf of my co-presenters at CAMBI. Prior BCI research suggests that neurofeedback may improve cognitive performance. As Alzheimer's is a disease associated with cognitive decline, including in non-memory domains, skills such as language and reading are often affected. The goal of this study was to develop a BCI-based neurofeedback paradigm to investigate the effect of neurofeedback on cognitive processes underlying reading for people with mild Alzheimer's disease. For this study, we recruited 6 participants with mild Alzheimer's disease. 2 participants completed the protocol in its entirety. COVID restrictions prevented the remaining participants from completing the protocol. The protocol was a non-experimental, multiple baseline, single-case research design consisting of 3 major phases: baseline, intervention, and follow-up. Each phase had sessions in which an experimenter went to a participant's home to assess BCI performance and cognitive measures. This procedure took place just once per week during the baseline phase, and all 3 sessions per week for 6 weeks during the intervention phase. All sessions included a BCI calibration and spelling task using the RSVP keyboard presented with BCI Pi, an open-source Python-based BCI software package. EEG data were collected with a dry electrode headset. Cognitive measures were repeated weekly to measure progress in all phases, including once per week on every 3rd visit during the intervention phase. The letter span task required participants to look at sequences of letters on the screen one at a time and repeat the letters back to the examiner. This task measured working memory. The letter cancellation task pictured here required participants to cross out a target letter. In the example shown, Z is the target, amongst non-target letters, as seen in the figure on the right. This task measured selective attention. The sentence reading fluency task required participants to read as many sentences as they could in 3 minutes while judging whether the sentence was plausible or not by circling yes or no. This task measured speed of processing. Parieto-occipital alpha rhythm was used for neurofeedback because of literature demonstrating alpha attenuation during tasks with mental effort. We found supporting evidence of this mechanism in a pilot study. In the pilot study, among participants who made errors on a one-back task, EEG frequency analysis revealed a 17% increase in posterior rhythm alpha power when participants made errors compared to when they made no errors. In the main study, we aggregated power spectral density data across baseline sessions in order to identify percentile cutoffs for level of visual feedback during the neurofeedback display. Looking at the figure on the right, the feedback levels range from dark green, which means excellent, to dark red, which means bad. Best to worst, the divisions between feedback levels were set as the 30th, 55th, 70th, and 85th percentiles of an individual's expected alpha PSD, where lower alpha PSD resulted in better feedback. You can also see from this figure that the feedback was presented after each 10-letter sequence. Most cognitive measures that were repeated multiple times before and during the intervention did not change significantly across phases. The figure on the left depicts area under the curve results by session for each participant. AUC for participant 1, the orange curve, changed about 0.14 from baseline to intervention. Participant 2, the blue curve, was more variable across phases with an AUC range of 0.55 to 0.85. The figure on the right includes examples where there was no change in performance with the introduction of the intervention, as in plot D, a sustained learning effect across baseline and intervention phases, as in plot J, a learning effect occurring only during the baseline phase, as in plot G, and improved performance after the intervention, as in plot A. The current study is proof of concept for delivery of neurofeedback via a BCI task for participants with mild Alzheimer's disease. There were limitations, including the sample size for this feasibility study was planned to be small, only 6, but due to COVID, only 2 individuals completed the intervention. The EEG for the study was obtained in participants' homes in order to decrease participant burden. Home recording may generate more and variable artifact compared to in a lab setting. As with all behavioral interventions, there could have been non-specific parts of the intervention that contributed to results, including aspects such as social interaction and cognitive stimulation. Future research is needed to investigate the extent and generalizability of these findings. Thank you for listening to our presentation. Please contact Fried-Oken with any questions regarding this presentation. Consider following our research group on Twitter at CAMBI Tech or on YouTube at CAMBI Research. Our website is also provided so that you can look at our other projects.